I'm a cell biologist, and my students and I work in the lab and ask questions about what happens inside of cells. So our work is kind of like a giant Sudoku puzzle, if you will. We kind of know some of the things that are already happening inside the cell from the research that's been done previously. So we know maybe protein A does one thing and protein B does another thing. And we're a little bit unsure of what protein C's job is. So then we can manipulate protein A or protein B or protein C in order to see what happens. It's just like doing a Sudoku puzzle. If you get all the pieces put together in the right order, you should be able to figure out where each thing goes, and that's what we do with cells. We have two big goals in our lab. One is just to understand the basic functions of the cell, and sometimes we don't know what we'll learn from that, and usually once we understand something, then we know, oh, this will help us make a cure or a drug or for a particular human disease. So most of what I do is biomedical research, and it has relevance for human health. The other thing in terms of the foam cell macrophages more specifically is to understand how those cells become dysfunctional and what processes mediate their ability to cause disease. The main one that's relevant to our research is heart disease, because we're understanding how macrophage foam cells get dysfunctional based on the lipid uptake and the changes that take place in the cell after it's taken up all of the lipid. And then the other thing that our research relates to is cancer. So one of the things that we study is growth factor receptors, and those are receptors that are on the surface of the cell. So those receptors can perceive a message from the environment or the area outside of the cell, and then they can actually relay that signal to the inside of the cell. People have sort of understood this basic concept for a pretty long time, maybe you could say a couple of decades. And so researchers have designed drugs. Once they've understood the pathway, they've gone and looked for drugs that would affect various components of it. And their assumption is if they can stop that signal, that relay that I was talking about, then they would be able to control the growth of the cell. But sometimes the thing they think will work, it doesn't actually end up working. So now we're understanding not only the different proteins that relay that signal, but also how they're organized in space and time, and that's where our microscopy techniques come in. So what we're understanding now is that it's not enough just to know what protein is doing what job and how the protein can get turned on to do its job or turned off, but also to know where it is inside the cell and how that affects its job. We have lots of students who are working on these questions, who are in the lab doing the experiments. And so part of what I see is my work is helping to answer biological questions, and hopefully someday it will make an impact on human disease. But I'm also really proud of the students and what they're learning and how they might take that knowledge and go out into the world in various ways if they go to medical school or graduate school or work in industry or whatever job they have when they're done.